Today we'll be focusing on another aspect of genetics which predominantly is termed as multiple alleles, and we'll be studying ABO blood group inheritance. And the basis for the ABO blood group inheritance is, in human beings, blood primarily is made up of antigens and also the blood plasma. As you understand, allele is the morphological expression of a gene, and any particular allele exists in two states. It could either be dominant or recessive. Normally, we have studied in Mendelian inheritance that alleles could either be in dominant or in recessive form, while in multiple alleles, alleles could be in different characters. They will have different characters. Alleles could have different characters, and the alleles could exist in the form of a series, like A is greater than B, B is greater than C, C is greater than C. Thereby, different alleles can occupy the same sort of a locus or the same sort of a position, and they could be expressed in different traits other than their A or B. So, this pattern of inheritance is predominantly termed as multiple alleles. A common form of multiple alleles is seen in the inheritance of blood groups. When you take blood groups, or when you take blood as a sort of a factor, we understand that blood groups is predominantly determined by it follows monogenic inheritance. That is, it follows single gene inheritance. And the most important thing is, when you take the single gene is designated as I. The single gene is designated as I. And I can exist in different forms, like when you study blood groups of humans, the possible alleles in the gametes of the father as well as the possible alleles in the gametes of the mother could be of different kinds. They could be A, or else they could be B, or else they could be O. You got three important types of antigens, A, B and O, which is present in human beings. Now, in today's class, we will be studying about how is the blood groups and what is the pattern of inheritance which is shown by I. We will now be discussing on blood groups and their genotypes. As you understand from the table, when you take a blood group, the blood groups could be A or B or AB or O. Normally when we take humans have A, B, AB and O. And the antigens, the foreign proteins which are present on the surface, the foreign proteins which are present on the surface of the red blood corpuscles could be A, could be B, could be both A and B, or in the case of the blood group AO, there are no antigens, there are none. And when you take the genotypes, they are predominantly determined by the I gene. The I gene can exist in different combinations, as seen by the crossing of a male gamete with that of a female gamete. So the genotypes which you could derive from blood group A is IA, as well as IA and small I. Small I primarily indicates which is from the O group. Small I is also designated as I to the superscript as O. Similarly, when you take blood group B has a genotypes capital IB, as well as IB and small I, or capital IB and also IO. Same is the case with AB. Now in the case of AB, both the genes, antigens A and B are expressed. So the genotype is determined by IA as well as IB. This is termed as codominance. So here in the case of blood groups, we understand there is dominance, again there is codominance, and the third one is it is determined by multiple alleles. Many alleles which are present in a series at the same sort of a locus, on a chromosome. And when you take O does not have antigens. It does not have antigens, and it is designated by the genotype small ii as well as IO. Many of the sort of a problems which will be solved is based on a recapitulation of this particular table of blood groups as well as the sort of a genotypes. Now we will be looking at the common objective bits which predominantly come in the frame of multiple alleles as well as study of blood groups. Now the first problem says, if you take Mendel's piece, one allele codes for which sort of a trait? An allele normally represents the expression of both the genes, that is both the dominant as well as the recessive gene. A pair of genes is predominantly termed as an allele. So in the first objective bit, we understand that when you say allele represents both round seeds as well as wrinkle seeds. Thereby the correct answer, as you see in the sort of an objective, is answer D, which says that you got rounded seeds as well as wrinkle seeds, both which are dominant and recessive, which you have read in Mendelism, and lethality or lethal genes are avoided. Thereby the correct answer is answer D. What exactly is the meaning of multiple alleles? When you take multiple alleles, it means if more than two alleles, like you think you got more than two sets, or you think of more than two people standing on at a particular sort of a position. If more than two alleles are present within a group of individuals a group of individuals who are occupying the same sort of a locus, it is primarily termed as multiple allele. So in a multiple allele, we have the situation wherein this is the locus, and we have a set of genes present over here. So we have a gene cluster. A gene cluster which is present at a specific sort of a locus, at only the same sort of a locus. And here you got two or more alleles. Two or more alleles which are present at the same locus, and the locus is the same, but the genes in the alleles are different. So this is primarily termed as multiple alleles. So multiple alleles is based on the position of the the number of the alleles and which occupy the same sort of position. So in the objective, we find that the choices are incomplete dominance. You have read regarding incomplete dominance in the last class, wherein a particular character is not expressed completely. Then you got dominance. Dominance is the expression of one morphological trait in a particular sort of a character, like only red or wrinkle. Then you got codominance, as you had seen in the case of the flowers. We had found that if we take red flowers and cross it with white flowers, we get pink flowers. That is codominance. Whereas in the case of multiple alleles, always in an objective we exactly find out what is the locus, the same locus, and how are the alleles. You got two or more alleles which are all clubbed together and present on the same position. So this is the concept of multiple alleles, and normal examples of multiple alleles are blood groups which you see in the case of human beings like A, B and O. Now we go to the third problem. In the third problem, we find whenever we encounter the term multiple alleles refers to a allelic series. We have an allelic series. That is, maybe we'll have A1, then we will have A2, then you will have A3, then you will have A4. This refers to a sort of an allelic series. So normally when you take multiple alleles, the number of genotypes possible in an allelic series is given as, in the formula as n. We have n is equal to n plus 1 divided by 2. N is equal to n plus 1 divided by 2, where n represents the number of alleles in the other locus, where n is the number of the alleles at the locus. Imagine we have a situation wherein we have two alleles. So it will be n2 is equal to 2 plus 1 is equal to 3 divided by 2. So the correct answer of the number of the genotypes which is possible in an allelic series is always given by the expression or by the formula n is equal to n plus 1 divided by 2. So this indicates the possible allelic series. So many times in a sort of a problem, they'll exactly be giving you, they'll just be saying the number of alleles at a particular locus could be 4 or 5 or 6 or 8, and immediately they'll ask you exactly how many types of genotypes are possible. So the number of the genotypes which is possible, the number of genotypes, how many genotypes, number which is possible, can be derived from the formula n is equal to n plus 1 divided by 2, where you understand that n is equal to the number of the alleles. So the correct answer for this particular thing is not given as n is equal to number of alleles is one choice, but the correct choice is n into n plus 1 divided by 2. It is not dependent on the Mendel's principles. It is also dependent on the Mendel's principle of segregation. So the correct answer to this primarily is n to the bracket of n plus 1 divided by 2. Fourth bit in the objective asks you that at a particular locus of a group means of a particular character, when you got more than two alleles, if more than two alleles are present at a particular sort of a locus, definitely there is an increase in the number of the phenotypes as well as the number of the genotypes. There is definitely the increase in the number of the phenotypes as well as the genotypes, because the more the genotypes, the more is the physical expression of the phenotype. It is not so the choice number A is correct, whereas B says is there a decrease in number. Whenever there are increased alleles, increased number of phenotypes and increased number of genotypes is the right sort of an answer. It is not related only to the phenotypes, it is not related only to the sort of genotypes, it is not related to the decrease in the phenotypes and the genotypes, whereas the number of alleles, the more the alleles, the more is the phenotype and more is the sort of a genotype. So the easy way to understand for you is, if the alleles are higher, automatically the phenotypes would also be higher, and basically the genotypes also tend to be higher. So this sort of a relation is a characteristic feature of multiple alleles. So whenever you take multiple alleles, all multiple alleles show increase in alleles is equal to increase in phenotype as well as increase in genotype. Thereby, for problem number 4, the correct answer is increase in the number of the phenotypes and the genotypes along with the increase in the, there is more than 2 alleles. If there is more than 2 alleles. The fifth question says, when you take an ABO blood group system, then how are the genes for the antigens? When you take an IA gene, an IA gene is the antigen is the gene for A. Now similarly, when you take IA and IB, they both are codominant. They both are codominant. They both are expressed. And when you take the combination of IA as well as when you take the combination of IB and I, you will find that there is a presence of 6 genotypes. There is always a presence of 6 genotypes. This is clearly shown in the sort of a printed chart. It is shown in the chart wherein the possibility of all the sort of a gametes is possible. Thereby, the first one, that antigen A is represented by IAIA. Then you got, if you take AB, it is represented by IA and IB. And again, we have got small I, IA, IB and also small I. They are represented by 6 genotypes in the pattern of inheritance of blood groups. So an objective bit could primarily be asking you like whether there are 6 genotypes or 5 genotypes or 4 genotypes, which is codominant and which primarily is dominant and what is not expressed. So you have dominance relationship, you got codominance relationship, as well as you got also multiple allele relationship, especially with relation to A, B, O blood groups in human beings. So this is a very important sort of a concept. The inheritance of, when you take inheritance of blood groups, you got 6 sort of a genotypes. This chart on the inheritance of blood groups primarily presents the combination between what are the possible alleles in the gametes of the father and the mother. So as you understand, if you take the checkerboard, in the checkerboard we find there is the probabilistic combination of either capital IA or IB or I. It could be represented as II or even I with a capital I or a small I. So we have a combination of capital IA or IB and small I from the parental side, as well as again from the maternal side we have the same combinations. So we understand there are 9 possible type of combinations as you see in this sort of a chart. In the first one, it could be either an IA, and in the second, while you go across on the first, in the first row across, you find in the first one you have an IA combination. Next you have an IA and an IB combination, and in the third you got an IA and a small I combination. When you come to the middle row, we have an IA, IB. Then again you got an IB and an IB, and again you got an IB and an I. The second combination where you have got an IA and an IB, it primarily deals with codominant. It is said to be codominant. That is, both the genes are expressed over here. And when you take at the lower end on the left, you got a small I. When the small I combines with an IA, as you understand, the phenotypic character capital IA is represented first. IA, then it is IA and small I. Then it is IB and small I, and then you got an I and I, which is a characteristic feature of the blood group number O. So the inheritance patterns, based on this particular patterns, you could be getting multiple choices, because they would be giving you the combinations and they'll ask you to sort of a check up with relation to how the fill goes on in the sort of a checkerboard. This is problem number 6, and in problem number 6, as you look, we find that when both the alleles of a gene express themselves in the heterozygous condition, we are talking about a heterozygous condition which you have recently seen, like as in the case of you got an IA and an IB, that is a heterozygous condition. So this sort of a heterozygous condition is also termed as codominance. So that is why for question number 6, the answer is B, which is codominance. It cannot be the phenotype of one parent which expresses in the F1 hybrid. And phenotype of both the parents is expressed but not the intermediate phenotype. It does not go that way. And MN is an obsolete method of classification of blood groups. So the right choice seems to be codominance. So in the case of the codominance, both the genes are expressed. Hence the answer B is correct for question number 6. As you understand, for question number 6, the correct answer seems here. Codominance. So we find that in codominance, you got an IA and an IB. So both the alleles of a gene express themselves in the heterozygous condition. So this is heterozygous condition. And you have both the alleles. You got this is one allele and this one is the second allele. Two alleles together expressed in the heterozygous condition, and you call this sort of a condition as codominance. Now in problem number 7, there are 4 important statements related to dominance, related to codominance, related to multiple alleles, as well as incomplete dominance. So at least there is a possibility that you could get about some 5 to 6 questions based on this particular concept. And this is a very popular sort of a bit which normally comes in entrance tests. If the phenotype of the heterozygote is the same as that of the homozygotes, if the phenotype of the heterozygote is the same as that of one of the homozygotes, like you take the situation of capital T, small t. So you got a heterozygote called capital T and a small t, and you got either a small t or either you got a capital T, which is from the sort of a parents. So the question number A, the match is 4, that is dominance. So in a dominance relationship, always the phenotype of the heterozygote is the same as one of the sort of homozygotes. Next we come to B, question number B. So here B bit says the phenotype of the heterozygote is intermediate or falls within the range between the two phenotypes. There is a blending. So whenever there is a blending or it falls in between, so both the genes are expressed. So this is primarily termed as codominance. Each gene, so this is seen in the case of blood group inheritance as in the case of the AB blood group of human beings. Phenotype of heterozygote is intermediate, falls within the range. So the answer here is B3, that is codominance. Then we go to bit say C, which says that phenotype of the heterozygote includes the phenotype of both the homozygotes. It includes both the homozygotes. So bit C primarily refers to, we are talking in terms of multiple alleles. In multiple alleles, you got a heterozygote and it includes the phenotype of both the homozygotes. All the things are present. You got a capital gene, you got a recessive gene, and again you got an intermediate gene. All of them are placed on the same locus. So the correct answer is multiple alleles for this. And if there is a presence of more than 2 alleles at a particular sort of a locus, if there is presence of more than 2 genes at the presence of a locus, that is D, then it could also be termed as incomplete dominance. So incomplete means, in a sense, it is not complete. Still there is a need for one more gene to get added up over here. So this was with relation to problem number 7. What exactly is a sort of a multiple allele? A multiple allele is one wherein a single gene can exist in different alternative states. So primarily it could exist in different alternative states, and again the single diploid sort of an organism you can always have more than 2 alleles only. The permitted number of alleles which are present on a particular locus is only 2. So if there are 2 alleles which are permitted at a specific sort of a locus, and again there are alternative forms at a particular sort of an allele, it is predominantly termed as a sort of multiple allele. So multiple allele, so the answer in this question number 8 is both choice number A as well as choice number C are correct, and thereby the correct answer is D. This is a very simple question. Like if you take parents whose blood groups are AB, what could be the blood type of the sort of an offsprings? The blood type of the offsprings for AB can be either A or B. So the choice remains as A. So just by looking at the particular chart, you exactly know what is the blood group is AB, and you know what are the antigens which are present. So definitely the choice could be either A or B. So thereby the correct answer is A. Question number 10. Children of parents with blood group of AB will have blood type A or B, B, O or AB, C, AB, D O, A, B or AB? The answer is, if the blood group of the parents is capital A and capital B, then definitely when you take the children can have A or B. Hence, the answer is E. We understand when you look at the particular problem, you understand a man with a blood group A, marries a woman with a blood group B. Their child has the blood group O. The child has a blood group O. Thereby, what could the question which is asked is, what could be the genotype of the parents? The genotype of the parents can be either IA small i, or it could be IB small i. Thereby, what happens is, the choice you can get is, you can get an IA, you can get an IB as well as a small ii. Thereby, whenever you're studying up problems related to crossing of parents, when you're looking at how could parents cross, it's very, very important to exactly know what are the genotypes. And once you know the genotypes, you got to look out what sort of things are present over here, IA1 as well as IB1. This is the first one, this is the second one, and the third, what other sort of choices left behind the third one. So a perfect analysis of the genotypes would exactly, from this you can easily deduce what could be the sort of a genotype of the parents. Thereby the correct answer is IA, then again IAI, it is IAI, because you also have a sort of an you have a smaller I coming over here. Then again you also have an IBI. Thereby the choices answer is B. Many times in the study of multiple alleles, an objective bit could also be asking you about the genotype of the parent. Like if you take problem number 12, it says the child has a blood group of O and the father has a blood type of it's got a blood group type B. Then what could be the genotype of the father? The genotype of the father could predominantly be, like wherever here we have IO, which is from one of the child, this belongs to the child, I or IO. So definitely, because the father has got blood group B, the choice remains as IB. And if one of the genotypes is IB, then the next choice is also IO. Thereby, the correct answer among the choices for the question number 12 is B, which is designated as IB and IO. The ABO blood group of humans is an example of, dominance, incomplete dominance, codominance, or multiple alleles? The correct choice is, it shows dominance because there is a single gene monogenic inheritance in the form of I. Then again there is codominance, as you see in the case of A and B. And again it's a example of multiple alleles because we have got a cluster of genes which come together at a particular sort of a locus, like you've got the A gene, then you've got the B gene and the O gene, and the genotypes which are expressed are IA, IB and I. Therefore, primarily for this particular choice, you understand that the choice is, it has got both dominance, codominance and multiple alleles. When you take the red blood corpuscles, in the red blood corpuscle cells, on the surface, on the plasma membrane, they have foreign bodies which are termed as antigens. The antigens mix with the antibodies which are present in the plasma and are responsible for the agglutination or the clotting of the blood. Now, for the antigens to form, it requires certain genes to react. So, in problem number 14, we say which genotype does not form any sugar polymer on the surface of the red blood corpuscles. So, if there is small I and small I, a recessive gene, an antigenic property is not expressed, thereby in blood group O, you do not have the formation of any sugar polymer, and the right answer is D, which is represented as small I, and the genotype is also designated as small I recessive. A look at the blood group typing. When you look at the blood group typing, if one of the alleles is contributed by IA, this can be seen from this particular chart as well as the combination chart of all the gametes. If one of the alleles is IA, definitely the another contribution is always IO, then the blood group is A. This is the sort of a blood typing. Normally, given the genotypes, they ask you what could be the sort of a blood group. If an allele is contributed to one of the parents is IA and the other is contributed as IO or II, what is the blood group? The blood group is A. So the right choice between A, B and O for question number 15 is A. This is a concept based question. You understand, when you take ABO blood groups in humans, it is controlled by a single gene. So the number of genes in the inheritance of blood groups is single, so it is termed as monogenic. How many types of alleles are present? Here we got three important types of alleles: IA, IB and small I are the three important alleles which are present in blood group typing. How many different alleles are present? You got three different alleles. And what could be the different sort of genotypes which you get formed? The different sort of genotypes which you get are predominantly 6, 1, 2, 3, 4, 5, and again one more gets added, it's called 6. So the question asks, how many phenotypes are present over here? The number of phenotypes that you get is predominantly, it is 4, it is not 3, it is 1, it is not 2, but predominantly you got 4 different sort of a phenotypes which is present. This can be studied by checking up with relation to the sort of a chart. So you should know the number of the phenotypes is always 4, the number of the genotypes is always 6, and there are 3 different alleles, and the 3 alleles are IA, IB and I. And it is the entire blood group inheritance is determined by a single gene which is termed as a monogenic. It shows a monogenic pattern of inheritance. The father has a blood group A which is unknown, that is the genotypes are not known. The blood group is A, the genotypes are not known, whereas the mother is B and the mother is homozygous. So given these three situations, like you have an A, genotypes primarily, father is unknown and B is homozygous, the question says, what is the blood group of the progeny? In the blood group of the progeny, we could have, because of A and B, the probability is AB, as well as again the probability is also B. B is also a sort of a possibility because B and B can combine together and it does not have the genotype of A. Thereby, the correct answer to this particular problem of number 17 is AB and B is possible, which is the right answer is A. We understand in an ABO blood group system, both the antigens are present in AB. We have antigen A as well as antigen B. But because both the antigens are present, there are no antibodies. So when there are no antibodies, the type of the blood group which gets formed is AB. So because both are present, that is, you do not have an antibody A and also an antibody B, both are absent. Both are absent over here, whereas both the antigens are present. Thereby, this is seen in the case of, especially in the case of blood group AB. Hence, the answer to the question number 18 is AB, that is the choice is C. In a multiple choice, often the genotypes of the parents is asked, given the sort of an offsprings and given the blood groups of the offsprings. So, question number 19 says the offsprings produced from a marriage have only O and A blood groups. Because offsprings have got only O and A blood groups, you understand blood group A, the genotypes could be small I, and blood group A, the genotypes could be IA dash as well as IA as well as small I. So the possibilities that the possible genotypes of the parents which predominantly can come when O and A are the sort of an offsprings is given as IAA, this one, IAA, as well as it could also be an IO and an IO together. So the combination of this is one of the gametes and this is, these two together can form the sort of a second sort of a gamete. So this could be the possible type of the parents. The possible parents could be from this. So many times the offsprings, the blood group of the offsprings are given and the genotype of the sort of parents are asked. So this sort of a matrix chart can exactly help you to study up with relation to the different patterns of inheritance of blood groups.